Hi Jenica, I've got a video for you on gene profiling and gene sequencing. So here you go. Here is some notes. So for both of them, we can extract the DNA from a cheek cell or a scene of a crime or something else. We can amplify it using PCR, which I think you're pretty okay with. So we would heat up the DNA to split it. We would use Taq polymerase to start adding nucleotides, and in between those two steps we'd add the primers at a lower temperature. We need Taq polymerase because it's heat resistant from thermophilic bacteria. So in our thermocycler we would need Taq polymerase, our DNA sample, and our free nucleotides all ready just to amplify the DNA, so we've got loads of it. And then we can do two things, but most of them require us to chop up our DNA into small bits. So we chop up our DNA using restriction endonucleases, which are restriction enzymes that have recognition sequences. So it might be that it recognizes ATCT. It would normally be a lot longer than that, but I've just simplified it. So we've got a mixture of restriction endonucleases will provide a section of DNA at different lengths. So we've got chop our DNA up into some different sized fragments, which we can then separate to form a profile via electrophoresis, which I think you're pretty okay with, going from negative to positive in order of size or weight according to the number of fragments. So we've also got sequencing, so we can sequence our DNA. So once you've got enough DNA, you might have already had enough straight away, but if you haven't, you'll have to do PCR, and then we would need to chop our DNA up into fragments using restriction endonucleases because we can't cope with big fragments in our machine. And then we have another thermocycler similar to the PCR machine, and in that machine we're going to add some DNA polymerase, which is probably Taq polymerase, but the book doesn't say. We've also got some primers, some free nucleotides, and some fluorescent or radioactive nucleotides, but not very many of those. And so we would add all of that together. Our DNA is broken down by heat. So there's one fragment there that's been broken down by heat. The primers are added so that our free nucleotides can start annealing to the single strand. And then occasionally one of these terminator bases that's been labelled will stick on the end of our chain, and as soon as that happens, that chain will stop replicating, so we don't have any more of it. And so we've got lots of different lengths, all the possible different lengths of DNA that we could possibly have, every single one. That continues until we've got all of those different lengths that you could come up with, and then those will be set in order in a capillary and detected by lasers and computers and other biotech magic. But that's the difference between sequencing and there's electrophoresis. The restriction enzymes are just used to cut DNA strands into smaller fragments so that we can do this or this. Hope that helps. See you Wednesday.